So chapter 9 is going to deal with the catabolic process of taking glucose and turning it into energy. And we do this using a process called cellular respiration. So all of our cells require energy to do work. Remember that energy is the capacity to do work. So the whole point of cellular respiration is to generate this energy. So we're going to generate this in the form of ATP. The energy starts stored in organic molecules. So it's going to start as potential energy in organic molecules, things like sugars. And through the process of cellular respiration, which is going to be many, many steps, we're going to eventually generate approximately 32 ATP molecules. So again, this is catabolic. We are breaking down organic molecules, which is an exergonic process. So this is going to release energy from those chemical reactions. When you think of the word respiration, what do you think of? If you think of respiration, you're probably thinking of breathing. So oxygen comes in, carbon dioxide comes out. And that's correct. So that's pulmonary respiration. We're going to talk about what happens to the oxygen and the carbon dioxide at the cellular level. This word aerobic means with oxygen. So aerobic respiration is when organic molecules are going to be consumed. So our body's going to break down some kind of organic molecule like a sugar, and oxygen is consumed. So it's going to require oxygen, and it's going to yield ATP. Anaerobic, so AN, when I add that to a word, means without. So anaerobic means without oxygen. So this is respiration. It's very similar. So it's going to consume compounds, but it's typically not going to use oxygen. A lot of times anaerobic respiration will consume something like sulfur instead of oxygen. This is common in organisms that live in oxygen deficient areas or really, really deep in the ocean near like hydrothermal vents. Fermentation is not a complete breakdown. So in aerobic and anaerobic, the organic components were consumed. That means that they were completely broken down into their components. Fermentation is a partial degradation. So that means that the sugars that we put into fermentation are only broken down a little bit, not completely. And this again is going to occur without. So there's no oxygen. The processes we're going to focus on here are cellular respiration first, and then we'll talk about fermentation at the end. So cellular respiration is our body's process of taking sugar and turning it into energy. There are aerobic and anaerobic steps. So the very first step is glycolysis, and this step does not require oxygen. So because it does not need oxygen, we consider it anaerobic. After that, we're going to have the citric acid cycle and oxidative phosphorylation. So the last two steps are both going to be aerobic because they're going to require oxygen. So cellular respiration is a mix of both anaerobic and aerobic. We usually just refer to it as aerobic because you can't do the whole thing without oxygen. You can only do the first step. So typically cellular respiration is often exchangeable or interchangeable with the word aerobic respiration. So down here we have a chemical formula, and I want you to think about the things that your body needs. What do you use for fuel for your body? How do you get energy into your body? You eat food. So one of the components of getting energy is the food that we eat. So this is glucose. So we're going to go through this whole process with glucose, and then we'll show you at the end how this can be different with proteins and lipids. We're talking about respiration, so that's breathing. So what gas do you breathe into your body? You breathe oxygen. So the two reactants required for cellular respiration are our organic molecule, which is our sugar, and our gas, oxygen. We'll have several steps to this reaction. So this arrow is going to be glycolysis, citric acid cycle, oxidative phosphorylation. At the end of all of these processes, we will release some byproducts. So one of the waste products that we release is carbon dioxide. And your body gets rid of this by exhaling it from your lungs. You're also going to make some water. This is not very much water, so we call this metabolic water. You might make about 400 grams of metabolic water in a day. Again, this is not enough to hydrate you. And the main goal of this is to create energy. So we'll create energy in the form of ATP, which is usable. And every step, according to the second law of thermodynamics, will also create heat that will just get lost to the atmosphere and won't be usable. You need to know this overall formula for cellular respiration. So you need to know that glucose and oxygen are the reactants, carbon dioxide, water, and ATP are the products. So as we go through this large metabolic pathway, we're going to see several redox reactions. So redox is a combination of two words, reduction, oxidation. So reduction and oxidation happen when we transfer electrons. Every time we move electrons, it's going to change the potential energy. So this is how we're going to get energy out of our reactants, is by moving electrons around. Oxidation occurs when you lose an electron. Reduction occurs when a substance gains an electron. So hopefully you can see how these are linked. If one substance loses an electron, it's oxidized. The substance that gained the electron is reduced. So you can't have oxidation without reduction. They're always going to come hand in hand. So we just shorten this to redox. So we might have two chemicals, so chemical A and reactant B. And what's going to happen is maybe an electron is going to be transferred from one to the other. Chemical A, who loses the electron, becomes oxidized. And chemical B, who gains the electron, becomes reduced. So we'll end up with A with a positive charge and B with a negative charge. There are some silly ways to remember this. One of them is Leo, the lion, says grr. When you lose an electron, you are oxidized, Leo. When you gain an electron, you are reduced, grr. The electron donor, so in this case, A is the electron donor because it had the electron and it gave it up, is called the reducing agent because A reduced B. The electron receptor, so this in case, the B is the electron receptor because it receives an electron, is the oxidizing agent because it oxidized A. I want you to focus on knowing what oxidation and reduction mean. So here's an example. We have sodium and chlorine, and these are going to come together to form sodium chloride or salt. So what happens is sodium is going to give an electron to chlorine, and it's going to form an ionic bond. So in this case, remember Leo, sodium loses an electron, so sodium becomes oxidized. Chlorine gains the electron, remember Gur. Chlorine gains the electron, so chlorine is going to become reduced. This is pretty clear to see when you have charged particles at the end. When we lose an electron, we get a positive charge. When we gain an electron, we get a negative charge. And so you can kind of tell who lost and who gained the electron. And so in a simple equation like sodium chloride, it's a little easier to figure out who got oxidized and who got reduced. When we look at the actual equation for cellular respiration, we don't see any overall charges. So we need to think about how the electrons are shared. We're going to start with the sugar, and the carbons in the sugar are going to get transformed into carbon in carbon dioxide. So we're going to look at the carbons in these two molecules. So in glucose, carbon is typically going to be sharing with hydrogen. We know that carbon and hydrogen have about the same electronegativity. This is going to be a non-polar covalent bond, and these electrons are going to be shared pretty much right in the center, equally between them. So they both have equal access to those electrons. When this process occurs, so from reactant to product, the carbons are going to go from sharing with hydrogen to now sharing with oxygen. Well, this is now a polar bond. Carbon is not anywhere near as electronegative as oxygen, so the electrons are going to be over here a lot closer to oxygen. So what happened to the carbon? The carbon went from having equal access to electrons to less access to electrons. So it essentially loses electrons. We can do the same thing looking at oxygen from the reactants to the products. So the oxygen that you breathe in actually gets turned into water. So oxygen starts out as oxygen sharing with oxygen, which this is again non-polar because oxygen and oxygen have the same electronegativity. So the electrons are going to be pretty much right in the middle, shared equally between these two molecules. When oxygen gets turned into water, now we've got oxygen sharing with hydrogen, and we know that this is a polar bond. Oxygen is much stronger, so the electrons in this pairing are going to be much closer to the oxygen. So the oxygen goes from having 50-50 equal access to electrons to actually getting more access to electrons. So essentially it is gaining access to electrons. It doesn't pull them completely away from hydrogen, but it does get to hang on to them a little tighter. So the oxygen is actually reduced from oxygen to water. So as we've seen many times now, when we do biological pathways and processes, we do lots of little steps and we pull energy out at every single little step. We're going to see that we have a very stepwise energy harvest. Sometimes the energy molecule is going to be really obvious and we're just going to generate ATP and you're going to know what that is. A lot of the steps in these pathways are going to create an energy transport molecule called NADH. So what happens is NAD+ is given some hydrogens plus the electrons that are associated with hydrogen, and it is reduced to NADH. So anytime that we move hydrogens around, we're also moving the associated electron around. So in this case, when we give NAD+ access to a hydrogen and an electron, it is gaining an electron and therefore it is reduced to NADH. NADH is a high energy molecule and it will store the energy for a temporary period of time, and then eventually the energy that is stored in here will go on to be made into ATP later on in the process at the electron transport chain. So you're going to see in the very first two steps, glycolysis and the citric acid cycle, we don't make a lot of ATP. We're going to make some, but we're going to make a lot of these energy molecules, NADH. And then there's another one that's similar called FADH2, which we'll make some of. In the very last step of cellular respiration, we're going to reach something called the electron transport chain. And it's literally very small steps down a pathway of electrons. And at every little step, we're going to be able to generate some ATP from the NADH. So the NADH will come in and donate hydrogens and electrons, and then we'll be able to harvest the energy from there. The molecule powering this process is oxygen. Oxygen's over here at the very end, and oxygen is very electronegative. So think about this as an electron magnet, and it is pulling these electrons down the electron transport chain. So we actually aren't going to be able to harvest any ATP without having oxygen present. And that is the whole reason that you have to breathe oxygen, is to power the electron transport chain. So in the next several videos, we're going to go through each of these processes in detail, and I will highlight exactly what you need to know about each of them. So the very first step is going to be glycolysis, and it is going to take glucose and break it down into 2 molecules of pyruvate. So glucose has 6 carbons. We're going to break these each down into 3 carbon molecules. This does not require oxygen. The second step is the citric acid cycle, which is going to take those pyruvate molecules and completely break them down into carbon dioxide. This one does require oxygen. And then oxidative phosphorylation, which is going to include the electron transport chain. And this, of course, does require oxygen. So we're going to make very little ATP in steps 1 and 2, and then the vast majority of ATP in step number 3. So as we go, we're going to track the number of ATP that are generated throughout these processes. So oxidative phosphorylation, that's that last step, is going to make the vast majority, about 90%. By the end, I will promise you, we will make 32 ATPs from each molecule of glucose. ATP is generated in a process called phosphorylation. If you think about that, we're literally taking ADP and adding a phosphate to it, so that's phosphorylating, to turn it into ATP. Sometimes we do this at the substrate level, where I literally pluck a phosphate off of something, stick it on an ADP, and turn it into ATP. This is going to be a very simple way to make ATP. We do this in the first glycolysis and citric acid cycle. The electron transport chain is not going to generate ATP in the same manner. It's going to use oxidative phosphorylation, which is going to require oxygen to pull electrons down the electron transport chain and then create a hydrogen concentration gradient and then pull the hydrogen through another protein to create ATP. So it's a much more complex method of making ATP. So the very first step of cellular respiration is a process called glycolysis. And glyco here means sugar, like glucose. And then lysis, we've seen several times now, means to break. So this literally means breaking glucose. So we are going to start with a molecule of glucose, and we are going to break it into 2 molecules of pyruvate. So think for a second, how many carbons does glucose have? Glucose has 6 carbons, and we're going to break that in half into 2 molecules. So pyruvate is going to each have 3 carbons. This is going to happen in the cytoplasm, so we are not entering the mitochondria yet, and it has 2 major phases. Just like any good investment, you have to put a little bit into it before you can get anything out of it. So the whole process of cellular respiration is to make energy. So we are going to have to put some energy in as an investment, and then we will eventually make a profit on energy in the form of ATP as well as some NADH molecules that will later be used to make ATP. Even though it produces a total of 4 ATP, we talk about a net production of a total of 2 ATP because it costs 2 in the beginning, and then we make 4, so we make a total profit of 2 ATP and 2 NADHs. Glycolysis is going to happen whether there's oxygen or not. Every single cell, whether it lives in an oxygen-rich or an oxygen-poor, whether it's a bacteria or a yeast or a human, is capable of doing glycolysis. Glycolysis is the starting point for all of the different pathways. Fermentation, cellular respiration, they all start with glycolysis. Later, we're going to use the mitochondria, but right now, we are not in the mitochondria. We are in the cytoplasm or cytosol. So the first thing we have to do is we have to take our sugar, so we have 6 carbons, and turn it into 2 times 3 carbons in the pyruvate. And we are going to make some ATP, 2 net, and we will make 2 NADHs. So here you can see the 2 phases of the glycolysis separated, and I like this image because it's going to let me highlight the things that I need you to know. So there are 2 phases. We can separate them with a line right here. So this is the energy investment. This is where we have to pay energy. So this is going to be negative. We are losing energy in the energy investment phase to get things started. And then the energy payoff phase here is where we're going to make our energy profit. So you need to know that as a reactant, we are going to start with glucose. And in the very first energy investment phase, we are going to lose 2 ATPs. So it's going to cost us 2 ATPs. In the second half of this process, we are going to make 4 ATP and 2 NADH. And the resulting carbon molecules are 2 pyruvate. And it's important here that there are 2 pyruvates. So down here in this net region, this is a summary of the different molecules. So we start with glucose, which gets split into 2 pyruvates. We make 4 ATPs, but we use 2 of them for a net of 2 ATPs. Be careful when you answer questions about how many ATPs are made in glycolysis. If I'm talking about total, we make 4 total. Usually questions are going to ask about net production of ATP, and that's usually 2. And we're also going to reduce 2 NAD plus into NADH. So an easy way to remember what we make in glycolysis is that there's 2 and 2 of pyruvates, ATP, and NADH for each one glucose molecule. So at this point we have done glycolysis. And that happens whether we have oxygen or not. Now the next step in this process is going to be decided by the presence of oxygen. So we can go one of 2 ways. If we have oxygen, we are going to move on to the citric acid cycle, followed by oxidative phosphorylation. If, however, oxygen is not present, then we aren't going to be able to do the citric acid cycle. Instead, we would move on to fermentation. So right now, we're going to assume that we do indeed have oxygen, and we're going to follow this left arrow here. After we've done glycolysis, we're going to move on to the citric acid cycle. So here you can see this in general in relation to the organelles. So glycolysis again happened in the cytoplasm. And now we're going to enter the mitochondria. So because we have oxygen, we're able to enter the mitochondria. So what is going to enter the mitochondria is the 2 pyruvates that we made in glycolysis, right? Because glucose is already broken down into pyruvate. Each of those pyruvates is going to get oxidized into a different 2-carbon molecule called acetyl-CoA. And then acetyl-CoA is going to enter this cycle called the citric acid cycle. And in the citric acid cycle, we're going to generate some more ATP. We're going to make some energy storing molecules like NAD and FADH2. We're also going to completely break pyruvate down into carbon dioxide. So we're going to lose a carbon dioxide here, and then we'll lose 2 carbon dioxides here. Remember that pyruvate has 3 carbons, 1, 2, 3. By the time we have lost those 3 carbon dioxides, the carbons from the pyruvate are completely broken down. Remember though that we made 2 pyruvates in glycolysis. So the entire process of pyruvate oxidation and the citric acid cycle is going to happen 2 times per glucose molecule, because it does 1 citric acid cycle for each pyruvate. Because we made 2, this whole thing is going to happen twice for each 1 glucose. So here's the first step. This is just a quick transition reaction that has to be done before pyruvate can enter the citric acid cycle. So here's pyruvate. We've got 1, 2, 3 carbons. And in this process, we are going to remove one of those carbons in the form of CO2. So you can see down here that the resulting molecule is going to only have 1, 2 carbons. So this is the first carbon we've lost. This process is also going to create a little bit of energy storage. So we're going to create 1 NADH. Remember that there's 2 molecules of pyruvate per glucose. So this whole thing happens times 2. So what that means is glucose started out with 6 carbons. In the pyruvate oxidation, we are going to lose 1 CO2 for each pyruvate. So we're going to lose 2 carbons total. And we're only going to have 4 carbons that actually enter the citric acid cycle. Each pyruvate makes 1 molecule of acetyl-CoA, and each pyruvate makes 1 NADH. So for a total of 2 acetyl-CoA and 2 NADH molecules. So we didn't make any ATP in this pyruvate oxidation yet. However, we have made 2 energy storing molecules here, which we're going to find out later are going to be able to take that energy and contribute it to making ATP. And then this acetyl-CoA is going to go ahead into the actual citric acid cycle. So next we have the citric acid cycle. It can also be called the Krebs cycle. So if you're looking and studying in a different place, you might see the word Krebs cycle. These are the same exact thing. And it is going to complete the breakdown. So by the end of the Krebs cycle, pyruvate and acetyl-CoA will be completely gone. And we will have generated all 6 of the carbon dioxides that we make from glucose. So again, this is a cycle, so it happens in turns, cyclical turns. So each turn of the cycle is going to make 1 ATP, 3 NADH, and 1 FADH per turn. However, the whole cycle happens times 2. So per glucose, we're actually going to make 2 ATPs, 6 NADH, and 2 FADH2. So this is just straight up energy. We know what ATP does. And these are going to be storing energy for later so that when we get to the electron transport chain, we can pull ATPs out of that chemical stored energy. So here's a quick summary. So to begin, remember we had pyruvate, which we're going to turn into acetyl-CoA. When we do that, we make an NADH and a CO2. The acetyl-CoA is then going to enter the citric acid cycle. In the very beginning, it loses 2 carbon dioxides. It's going to generate 2 NADHs over here. Here's my 1 ATP, 1 FADH2, and then here's a third NADH. So all of this, the pyruvate oxidation and then the citric acid cycle, is going to happen times 2 for each glucose molecule that we start with in glycolysis. So I don't need you to memorize all of the specific steps. I know that the book does go into every single step. However, the molecules that I need you to know are that we start with pyruvate. We lose NADH and CO2 to make acetyl-CoA. We lose carbon dioxide and NADH. We make ATP, FADH, and here's that third NADH. So I want you to be able to track the reactants that go into these processes and the products that are produced as they occur. So at this point we have done glycolysis and we have done the citric acid cycle, which means that we do have oxygen present in the cell. And by the time we've done the citric acid cycle, glucose has been completely broken down into 6 carbon dioxides. So we don't actually need this organic molecule anymore. So there's nothing else to actually break down. We've made several ATPs. So we made 2 ATPs in glycolysis, and we made 2 ATPs in the citric acid cycle. Remember that I promised you 32 ATPs, and we have now made a net total of 4, and so we need 28 still. And those are all going to come from oxidative phosphorylation. And the place that we're going to get the energy for this oxidative phosphorylation is the NADHs and the FADHs. So what we should have right now is we should have 2 NADH from glycolysis. We should have 2 NADH from the pyruvate oxidation. We should have 6 NADH from the citric acid cycle, and 2 FADH2 also from the citric acid cycle. So we actually have a lot of energy storing molecules here. So we have a total of 10 NADH and 2 FADH2. And these are all going to go into the electron transport chain to power the production of those last 28 ATPs. So this is going to happen inside of the mitochondria. So it happens on the inner membrane called the cristae. And if you remember what the structure of the mitochondria looks like, the inner membrane is highly folded. So this is highly folded to increase the surface area for the electron transport chain. For the most part, the electron transport chain is just a bunch of proteins. So they're just some membrane proteins all in a row, and they're literally going to pass electrons from one to the other. As the electrons are passed, they lose free energy. That means they have a negative delta G. So this is spontaneous, and they are going down in energy. When you go down in free energy, you are going to release energy. And that energy is going to be used to power movement of molecules. You'll notice that every time an electron moves, we're going to have a redox reaction because the one that is donating the electron is going to be oxidized and the next one in the chain is going to be reduced. So we often call this a series of redox reactions because that's literally what's happening. We're just passing electrons from one protein to the next. So here's our kind of overall image. So glycolysis happened out in the cytoplasm. We turned glucose into pyruvate. We made 2 ATPs and 2 NADHs. The pyruvate oxidation is going to produce 2 NADH per glucose and 2 carbon dioxides. The acetyl-CoA then enters the citric acid cycle, where we made a total of 6 NADHs and 2 FADH2, and a total of 2 ATPs, and we released 4 carbon dioxides total for the glucose. So there's 2 from here, 4 from there. That's all 6 carbons from the glucose. That's completely broken down. All we have left is our NADH and FADH molecules, which are all going to move into this inner membrane of the mitochondria to go through this process of oxidative phosphorylation. And then this is going to produce 28 ATPs. So here is a free energy diagram of the electron transport chain. I do not need you to memorize the names of all of the proteins. If you go to medical school, you'll learn them all then. So if you want to learn the names of the proteins, that's fine. But what you need to know is that the beginning of the chain has a higher free energy, and the ending of the chain has a lower free energy. So that means our delta G is negative. So we are going to release energy in this series of exergonic pathways. All the way at the bottom, we have oxygen. Oxygen is the most electronegative. This is what is called our final electron acceptor. Oxygen is going to receive electrons at the very end. When you receive an electron, you become negatively charged, which is going to attract positively charged hydrogens and is going to allow the oxygen to turn into water. So the very last step in this entire process is making water. So let's follow the electrons in this electron transport chain. The electrons are being donated by NADH and FADH. Remember, we've got 10 of these and 2 of those from the previous 2 steps. So NADH is going to give electrons to the first protein, and these electrons are then passed down this chain from each protein to the next. Every time an electron is passed, it releases a little bit of energy that can be used to do work. Generally, NADH is going to make approximately 3 ATP molecules, and that's just because it enters here at a higher free energy. FADH, you can see, enters at a little bit lower. FADH is going to generally make approximately 2 ATP molecules. Keep in mind this is approximate. There's a lot of other things that can affect how much energy actually gets pulled out of these molecules. So FADH and NADH do the same thing. FADH is going to give electrons to these electron transport chains, and then they're going to be passed down. The electrons eventually get passed to oxygen, which will combine with hydrogen to make water. And that is when the electrons exit the whole cellular respiration system. So this right here is focusing on just the movement of electrons. So electrons move from NADH and FADH2 to the electron transport chain. Eventually, they are given to oxygen. This is why oxygen is required. If oxygen was not there at the end, none of this would go. So none of this would happen because oxygen is at the very bottom, kind of pulling everything down like a magnet. All right, so we said that every time the electrons move, they release a little bit of energy. That energy is used to do cellular work. It is not ATP yet. So the energy is actually used to do active transport for hydrogen ions. So this membrane here is the cristae, that is the inner mitochondrial membrane. Inside of there is the mitochondrial matrix. And then this space is the intermembrane space, which is between the inner and the outer membranes. So here is kind of a simplified view of the electron transport chain. We can see the yellow arrows being the movement of electrons. And when each electron is passed, the energy is released to move a hydrogen from an area of low hydrogen ion concentration to an area of high hydrogen ion concentration. That is active transport. That's what the energy is being used to do. So every time an electron moves, free energy is released, and that free energy is going to pump hydrogens across this membrane to increase the concentration of hydrogen ions in this intermembrane space. So what's going to eventually happen is we are going to build up a concentration of hydrogen ions in the intermembrane space. So now our energy is actually stored in the hydrogen ion concentration gradient. And so once the hydrogen ions find a protein channel that is going to allow them to diffuse back down their concentration gradient, they're going to go through that. And the protein that does that is called ATP synthase. Think of this word synthase like synthesis. So it literally means ATP making. So the hydrogen ions are highly concentrated in here. This is a hydrogen ion channel that is going to allow the hydrogen ions to diffuse. The process of hydrogen ion diffusion through ATP synthase is going to create my approximately 28 ATPs. So this whole process is kind of broken into 2 parts. The first part is the electron transport chain, where we pass electrons and move hydrogens. The second part is where we actually diffuse the hydrogens to make ATP, and this is called chemiosmosis. Osmosis is passive transport. I'm not sure why they call it chemiosmosis. It's probably just because it's moving hydrogens and not water. You could just call it diffusion if you want. But chemiosmosis is this process of hydrogen ions diffusing through the ATP synthase and producing ATP. The whole process together, both of them, is what we call oxidative phosphorylation. So a lot of the time, these three terms kind of get interchanged, and if you mix those terms up, that's not that big a deal. I do the same thing all of the time. So let's follow the energy. The energy starts in NADH. As the electrons are moved, the energy then is stored in the hydrogen ion concentration gradient. Eventually, that concentration gradient is going to move through ATP synthase, and it's going to store the energy in ATP. So it's not a direct link from NADH to ATP. We first have to store the energy in a hydrogen concentration gradient, and then we can use that gradient to power the production of ATP. Here's a little bit of a closer look at the ATP synthase. So this is ATP synthase. It is a channel protein. It looks kind of like a rotor here. As the hydrogens move through, it turns this rotor, and the rotor makes ATP. So this is that process of chemiosmosis, where we have a high concentration of hydrogen on the inside and a low concentration of hydrogen in the matrix. And so this is going to be passive chemiosmosis, motion of hydrogen ion. So we often call this a proton motive force. Again, proton just means H+. Motive is our motivation. So it is a proton or hydrogen ion motivated force to do work. So here we can track the energy, or the chemical and potential energy that is stored within these molecules. It starts in the chemical bonds of the glucose. And then through the first two steps of glycolysis and citric acid cycle, we produce NADH as well as FADH2. Those then pass the energy to the electron transport chain, which is going to do those several stops in reduction of free energy. As the electron transport chain progresses, it creates a concentration of hydrogen ions. That's the proton motive force. And then eventually they go through ATP synthase to create about 28 ATP. Remember that the second law of thermodynamics said that no energy transfer is 100% efficient. The entire breakdown of energy from glucose only gets about 34% of the energy stored in glucose into ATP. So the vast majority of the energy stored in a glucose actually gets lost as heat, because we're doing so many reactions that each reaction is going to lose some energy as heat. So down here you can see the kind of total accounting. Glycolysis is going to make a net of 2 ATP. The citric acid cycle makes a total of 2 ATP per glucose. And then oxidative phosphorylation, which consists of the electron transport chain and chemiosmosis, makes about 26 to 28 ATP. And this is an approximate number, again, because this is a very indirect process. It's not quite as simple as just plucking a phosphate and sticking it on an ADP. Because it goes through so many steps, the number of ATPs can actually vary a little bit. And there's three reasons mainly why. Because it's not directly coupled, so because it has to go through the series of redox reactions, the ratio of NADH to ATP is not actually a whole number. It's approximately 3, but it's not exactly 3. The number of ATP varies, whether it's an NAD plus or an FAD in the mitochondrial matrix. So NADH is going to provide more energy than an FADH2. And then because the proton motive force is used to drive some other types of work, sometimes the energy in the proton force doesn't go directly to making ATP. It can go to doing other work inside of the cell. So we've done cellular respiration, which was the whole process of glycolysis to the citric acid cycle and then the electron transport chain. But all of that required oxygen to occur. Glycolysis does not. So glycolysis happens without oxygen. But without oxygen, you cannot enter the citric acid cycle or the electron transport chain. We can only do glycolysis. So when oxygen is not present, the products of glycolysis, which are pyruvate, will instead go through a process called fermentation. Remember in the process of glycolysis that we are taking glucose and turning it into pyruvate. During this process, we are going to make a net of 2 ATPs, which is great. We get some energy. During this process, we also take 2 NAD+ and turn them into 2 NADH. Normally, during aerobic respiration, these NADHs would go over to the electron transport chain, remove the electrons, and then the electron transport chain would give back the NAD+, and glycolysis can continue to work. However, when oxygen is not present, the NADH does not go to the electron transport chain, and NAD+ is not regenerated. So if we never actually make any more NAD+, this becomes a limiting reactant, and glycolysis is only going to continue as long as the cell has some NAD+. Eventually, this will run out, and we will have to stop doing glycolysis. That's really bad because once we stop doing glycolysis, we can't make any more ATP, and at this point, the cell can't generate any ATP. So the whole point of fermentation is to couple with glycolysis and regenerate NAD+. So the NADH is going to come down here to fermentation, and it's going to send back NAD+, so that the process of glycolysis can continue to occur. There are 2 types of fermentation. The first is alcohol fermentation. This is used by yeast, some bacteria. This is actually the process that we use to make alcohol, so like beer and wine, as well as baking, because the bread rises from the carbon dioxide released from the yeast. So the first step is glycolysis. Glycolysis, you guys already know, is going to make me 2 ATPs, 2 NADH, and 2 pyruvates. The pyruvate is then going to lose some carbon dioxide. This is why beer and wine have a bubbliness, because they've released carbon dioxide. The pyruvate is eventually turned into ethanol, which is an alcohol. And in that process, we are going to use the energy in NADH to transform it into ethanol, and that's going to regenerate NAD+ to continue to be used in glycolysis. So as long as this process has glucose, it can continue to make ATP and ethanol and continue to do glycolysis. So the whole point of the fermentation section is to regenerate NAD+ so that glycolysis can keep making ATP. A very similar reaction happens in humans. So this is called lactic acid fermentation. And what it does is it produces lactic acid or lactate instead of alcohol. Again, the same first step, we have glucose. We're going to make our 2 ATPs, our 2 NADH, and our 2 pyruvates. The pyruvate doesn't lose any oxygens or carbons. This is why we don't have carbonation or bubbliness in our fermentation. So this is going to happen when there's no oxygen around. Often this is happening during strenuous exercise. This might cause you to get kind of a cramp in your side. So what happens when there's no oxygen available to your muscle tissue is that they will go through this lactic acid fermentation. They produce lactate, and at the same time, we regenerate NAD+, so that glycolysis can keep making our measly 2 ATPs. Eventually, after you stop exercising and you start breathing again, the liver will turn lactate back into pyruvate later on and allow pyruvate to go over to the citric acid cycle and the electron transport chain. But both of these processes, cellular respiration and fermentation, all start with this process of glycolysis. So the very first step, no matter where you go, is to do glycolysis, make your 2 ATPs, your pyruvate, and your NADH. NAD has to be oxidized, or NAD is reduced into NADH, and this is a temporary energy storage. This has to be turned back into NAD plus for glycolysis to continue. This can either be done in the electron transport chain if you have oxygen, or it can be done with fermentation if you don't have any oxygen. There's a lot of differences between cellular respiration and fermentation, though. So they're going to have different ways of oxidizing NADH back to NAD+. In fermentation, we're going to have an organic molecule, such as pyruvate, turning into either lactic acid or alcohol. In cellular respiration, this happens at the electron transport chain. And there's a vast difference in the number of ATP produced. So fermentation only produces the 2 ATPs in glycolysis, whereas cellular respiration is going to create 32 ATPs because it can make a lot of NADH and then send those all to the electron transport chain to make a ton of ATP. An obligate anaerobe. Think about the word obligated. Obligated means you have to do it. So an obligate anaerobe is somebody who cannot survive in the presence of oxygen. So this is an organism that has to live with no oxygen. So these are always going to do either fermentation or anaerobic respiration. Sometimes they can do the whole respiration cycle and just use like sulfur instead of oxygen. There are many organisms like yeast and bacteria that are called facultative anaerobes, meaning that they can do either. They can either do cellular respiration if oxygen is present, or if you stick the yeast in a barrel and don't allow them to get any more oxygen, they will ferment and go through fermentation and make you alcohol. Humans are obligate aerobes. So we have to have oxygen. We can only do lactic acid fermentation for a very short period of time. Lactic acid builds up in our muscles. It causes problems. It's actually a metabolic poison. So if we stop breathing oxygen and we stop doing cellular respiration, we die. So because glycolysis is that very first step in all of these reactions, we believe that this is the most ancient process. So we likely think that glycolysis formed first, that early prokaryotes were able to do glycolysis before oxygen even accumulated in the atmosphere. So early planet Earth had a lot of carbon dioxide and not very much oxygen until later on when plants started to develop on the planet. So it's likely that glycolysis is a process that formed on early Earth before even plants were around, because it would have been able to function and make some energy without oxygen. It's then probable that other additional organelles, as they developed, were able to kind of add to this pathway to create this whole process of cellular respiration, which includes the citric acid cycle and then the electron transport cycle, which are much more complicated metabolic pathways. So this whole time we've been looking at just glucose. But the truth is that other molecules can be used for food. We know that all carbohydrates can be used for energy, so starch, glycogen, disaccharides like maltose, sucrose, and other monosaccharides like fructose can be used for energy. And those actually are going to just enter glycolysis. So something like starch probably just needs to be broken down into glucose. Same thing for glycogen, it just needs to be broken down into glucose, and then it can go right into glycolysis. Proteins are usually not used for fuel except in a very, very extreme starvation situation. So most of the time you're not using proteins for energy unless you're starving yourself. However, they can be. It's not very fuel efficient, but they can be. So the amino acids can actually lose their amino groups. So we're going to get rid of the NH2 because we don't want the nitrogen. And then this is going to enter into the citric acid cycle. Fats, remember, fats have the most calories per gram. So fats actually have a ton of energy stored. The glycerol is broken down into a product called G3P, which is actually one of the intermediate steps of glycolysis. And then those fatty acids can be broken down into acetyl-CoA, which can then go into the citric acid cycle. We know that fat produces more than twice the amount of ATP as carbohydrates or proteins. So fat is going to produce about 9 calories per gram, and then carbs and proteins only produce about 4 calories per gram. So fat is a highly energetic energy storage molecule. So here you can see kind of where those enter. Carbohydrates are going to either enter as glucose, meaning like we take starch and enter it as glucose. Some other ones like fructose. The second step of glycolysis is actually to turn glucose into fructose, so they can enter usually in this first step of glycolysis. Fats, which are our next main source of energy, the glycerols can be turned into something called G3P, which is a step in glycolysis. And those large fatty acid carbo chains are typically broken into 2 carbon chains that can be turned into acetyl-CoA and then go through the whole citric acid cycle. Amino acids need to lose their amino group, so they need to get rid of that nitrogen. They can be turned into a variety of different things, so pyruvate, acetyl-CoA, or more often, one of the citric acids put in the citric acid cycle. All of these processes are entering in either glycolysis or the citric acid cycle, which means they're going to help produce NADH and FADH2, which are going to be able to come over here to oxidative phosphorylation and make a whole lot of ATP.